Okay, now moving along to section 17.7, recombinant DNA, we are going to be talking about genetic engineering, things that we can do to cut DNA fragments and reassemble them at the way we want to. Also, we can insert human genes into DNA of bacteria to produce, well, one that we're going to see is insulin. And we're going to be basically talking about what's called the polymerase chain reaction. And also, well, first we're going to talk about recombinant DNA, when the polymerase chain reaction is going to use a similar type of something like recombinant techniques in its applications. But we're going to start with the recombinant DNA. Okay. In preparing recombinant DNA, you use a restriction endonuclease, it's called, a restriction enzyme, to remove a gene from a foreign DNA and also open DNA plasmids in Escherichia coli bacteria. Now, a restriction enzyme recognizes an enzyme, a specific sequence in the DNA, and it will cleave the DNA at that point, and where that sequence shows up. So you can have that happen in, you can have this particular sequence in human DNA and also in E. coli DNA. So you can use that restriction enzyme to open those genomes. In this case, well, in the case of E. coli, you're using the, you're going to take a piece out of the E. coli, and then you're going to replace it with the piece you cut out of the human genome. Okay, and you can do this with other species as well. And they talk about DNA plasmids, which a plasmid is a small circular piece of DNA that shows up in E. coli. It isn't necessarily the main genome, but these little circular pieces are easy to work with, and you can isolate them, you can cut them, and you can put other things in them. So what you do is you do that, you take a piece out of the E. coli plasmid, and then you replace it with a piece that you cut out of a genome that has a gene that you want to express, and you react these with a ligase, which then puts these pieces together, and when you see the diagram, it'll become clearer. So then you have a new gene in the altered DNA, and it produces a protein which, if it's insulin, you can isolate that a lot more easily, and you can actually produce a lot more of it than if you were to do what they used to do and use it, isolate it from insulin from, I think it was pigs' pancreases, and just the supply of insulin. And this is, first of all, it's going to be human insulin that's going to come out of this process, and second of all, you're going to have a much larger supply of it potentially. And here's a diagram of what's going on. You see the DNA plasmids are these small circlets of double-stranded, double helical DNA, and you can isolate them, and then you open them up with a restriction enzyme. You see they're taking a piece out there, and they have what these call these sticky ends. And the sticky ends, of course, are going to be ends that they are not paired with another nucleotide, and they can be paired with the nucleotide which has, they can be paired with another sequence of DNA, a strip of DNA, a length of DNA that has complementary sticky ends. So you get these sticky ends. You see you have a gene obtained from a donor DNA in green here. So you use the same restriction enzyme, so it cuts the chromosome at the same point, or at the same sequence, which means these sticky ends are going to be complementary to the sticky ends in the the part of the plasmid that you're going to try to splice this into. Okay, so then you incubate these by proper procedure, and you have a ligase. It's an enzyme that is going to join these bonds. You may remember, or should remember from the last chapter, chapter 16, that a ligase is something that's going to join two molecules together. So it's going to tie things together. And that's what it does. And then so you do this, and what you now have is an E. coli plasmid with recombinant DNA. The recombinant DNA, and you've taken a gene that you want to make the protein from, and you've put it into a DNA plasmid. Now you can just keep growing these things and make lots and lots of this stuff. And that's how that works. And here's some of the things that they do with recombinant DNA. And I'm going to mention too, I sometimes talk about the old days. Back 50 years ago, we knew of recombinant DNA. We kind of knew that this kind of thing happened, and there was talk of doing things like this, but it hadn't really been done, at least not at any commercial level. So as you see, we've come a long way in the last few decades. We do all kinds of things. There's a lot of things you can do with recombinant DNA, and I know there's controversy over it. Some people really don't like the genetic engineering that results from it, but that's not an issue to worry about here. Okay, another wrinkle in this is the polymerase chain reaction. Now, this is very useful. You can amplify, well, you can make multiple copies of a DNA, lots of multiple copies of the DNA in a short period of time. And what you do, you find a region of DNA you want to copy, and you're going to have a DNA sample from a crime scene maybe, or if you're trying to establish paternity or some genetic testing of various kinds, if you're looking for this, what you do is you have a strand of DNA that you want to copy, or a region of DNA that you want to copy, so you're going to have to isolate it somehow. Then you heat it to, I think it's 95 degrees Celsius. It's not in here. There's a biochemistry course that I watch lectures from to kind of get ready for some of this. It's 95 degrees Celsius. DNA is pretty stable. 95 degrees is going to denature it. It's not going to really change the sequence. It's going to come back together eventually. What you want to do is you want to separate these, and then you mix the separated strands with, it says, enzymes and nucleotides, which form complementary strands, and then you do it again and again and again. It's done a number of times over. Depends on how large a sample you want, but you keep repeating this. So you have this little piece of DNA, you heat it to separate it, and then you react it with enzyme, the appropriate enzymes and nucleotides to make this DNA. And remember, you don't need to transfer RNAs or anything like that. We're not making a protein here. We're just making DNA. And you just keep doing this over and over again, and sooner or later you have quite a lot of the stuff. So you can screen for defective genes. You can defective genes, genes associated with breast cancer as an example. And then they mentioned there's 2 known breast cancer genes that correlate to a higher risk of breast cancer. There's actually quite a few genes that correlate to some kind of possible disorder. And this is what you do with these things. In part, is you're going to you might cut this in various different ways and run it through electrophoresis and see the pieces, how the pieces move compared with a controller. Well, it'll be clear in the next couple slides. You have DNA fingerprinting. So you take a restriction enzyme, cut the DNA into smaller fragments, and then what you do is you separate those fragments using electrophoresis, and you can react this with, you can do various things to this stuff. You can use fluorescent light, or you can incorporate radioactive things into the radioactive elements into the DNA. And there's a lot of ways. What you're doing, you see what the scientist doing there is, or technician scientist, he's looking at the patterns of these various pieces cut from the DNA, and any two individuals are going to have different patterns, and you can establish paternity. You can identify you can identify that somebody was at a crime scene, things like that. So the DNA fingerprint is unique to each individual, and there's they give in the book here. It's the DNA fingerprinting. They base this on a set of 13 hereditary traits. The chances of two people having the same traits, unless they're identical twins, is something like 1 in 575 trillion, and that's in your textbook. So that's one of the things that this will do. And then the Human Genome Project, and I know I mentioned 6 billion bases. I mentioned 6 billion bases somewhere, and I think it was in one of these presentations. But they're saying now that it's 3 billion bases, and I think I also mentioned somewhere that the total number of bases in the human genome, the estimates have kept going down over the years. But the 3 billion bases represent, well, this is what was done in 2003, the Human Genome Project. There were 21,000 genes coding for protein. So we have essentially 21,000 proteins in our body, or genes for 21,000. Represents only 3% of the total DNA. Now, I might have also mentioned at some point that years ago, we pretty much knew that only a small percentage of the DNA coded for genes, and there was said, well, it doesn't really, the rest of it doesn't have any function. And other people said, well, we don't know what the functions are. And as it turns out, the functions of the DNA is regulatory. There's recognition sites for proteins. There's regulatory things. There's what they call telomeres, which aid in DNA replication, and they also shorten the DNA, which is kind of leads to a sort of programmed cell death, which is why we can't keep living forever, because sooner or later our somatic cells divide a certain number of times and they're no longer capable of it, and they don't reproduce. They die out. So which is why we're not immortal. Bacteria sort of are. I mean, they can keep dividing forever. But anyway, there's I see something here that probably would be better if I made it a little differently. There, that makes sense. So anyway, much of our DNA regulates genes. It serves recognition sites for proteins. And there's one other thing I didn't mention in the DNA. Some RNAs, some genes have in their sequences in DNAs, they have these little introns, I guess they're called, where there's segments that don't really code for anything and then have to be removed they have to be removed from the RNA, the messenger RNA, in order for, and then They have to be removed and then the rest of the RNA put back together so that these things can actually make proteins. So there's quite a few things that go on here. And obviously that this subject could serve as, you could do a whole course on that. So we're not going to go into any great detail at that point. So that's section 17.7. We're close to the end here. So let's do a learning check. Recombinant DNA technology requires the use of. Now, if I were in class and I showed this, there had better been several people who would pipe up and say restriction enzymes and plasmids, because that's what recombinant DNA uses. That's what we talked about, recombinant DNA. There's just that's the only possible answer there to that one. And you see restriction enzymes and nucleases. Okay, we are done with 17.7. I'm going to get this up and get this up to on Facebook or YouTube. I'm sorry, and you can have a look at it. And we'll finish up with section 17.8.